Today I'm going to try to convince you that if you care about longevity, it's worth considering spending your time in figuring out how to develop better genetic medicines. And the reason why is that there's a huge amount of effort being put in to finding genetic targets that are important in aging. People are doing high throughput screens, both in vitro and in vivo, using combinatorial perturbations to our genetics. People are looking at large scale sequencing cohorts, both of like supercentenarians as well as just like the general population using the UK Biobank and all of us data sets, where they do whole genome sequencing of hundreds of thousands to millions of people, and then you can use that to try to interrogate what genetic changes lead to greater or less healthy aging. People are finding isolated subpopulations with unusual phenotypes, companies like Variant Bio, where they're finding these populations, sequencing them, and trying to find the genetic causes of those unusual phenotypes. And then also people are trying to find reprogramming factors. There's many companies trying to find what combination of transcription factors in the right dosages and timing can shift our cells' phenotypes from an aged phenotype back to a younger one. So all those people are working on finding genetic targets, and that is like very important. We don't have any good ones for aging yet. It would be nice if we can find them. But there's not as many people working on the second part, which is that even if we have a great genetic target, you then have to turn that into a drug. And I read some things where people are like, oh, that's like already solved, but that is like definitely not the case. There are 7,000 or 9,000 genetic diseases where we know with 100% certainty that this genetic change results in the disease, and we have effective treatments for maybe half a dozen to a dozen of them. And so there's a huge amount of progress that needs to be made on developing genetic medicines, so that way we can take all these genetic targets and perturbations that people find and turn those into medicines. And so what are some possible things that you could work on if you are interested in that? One is that the genetic medicines that we have right now, they often don't go into that many cells, they don't go into the right cells, and they don't produce enough of the genetic effect that we want. For example, there are AAVs, the gene therapies that go into the liver. They only edit like 10 to 20% of your hepatocytes, and then their effect decreases over time as your hepatocytes turn over and your cells react to this foreign DNA. And so if you look at the amount of protein being produced, it's just on this like straight decline that will make them not that useful and not that long. You can't redose them because we get antibodies to the AAVs, and then if you redose them, your immune system just takes care of them. So there's a whole bunch of things we can do just to make the genetic medicines more effective. And this is true for all the genetic medicine technologies that we have, whether it's like prime editing, or it's mRNA therapeutics, or it's what else can we do? I mean, there's a wide range of genetic medicine technologies that are still not that good. Another key bottleneck is delivery, that we are really good at delivering these things to the liver, because your liver is where if you put something foreign in, its job is to take care of it, so lots ends up there. But there's many other cell types that we probably want to affect. It's not going to do you much good if you just have like a super healthy liver and then the rest of your cells are aging just as fast as normal. So figuring out how to do delivery to other tissues, or even to the liver, we're still actually not that good at it, depending on the technology. And so yeah, delivery is another key problem that people can work on. Third is figuring out how to drug specific genes using small molecules. That most genetic medicines right now, they're nucleic acid therapies, which are amazing and very effective for certain things, but it's hard to get nucleic acids into all the cells in your body. They're often complicated to dose. If you want to do genetic medicines in the brain, for example, you have to inject it directly into the CSF, which is not the most convenient. And if we can solve that using better small molecules, I think that would unlock a huge amount of like practical drugs for people to take for these things. People are using AI methods to try to find these small molecules. There's a bunch of work in macrocyclic small molecules that potentially have enough specificity. There's a lot you can do there. And so and the other thing is, if you're interested in founding a company, is that if you are able to make progress on any of these things that I mentioned, you can easily get biotech funding. You can make money sooner by treating one of those 9,000 genetic diseases, or many of them, before we find the aging targets to go after. So yeah, that's my talk. Work on genetic medicines. They're not very good, and if we can greatly improve them, you'll both treat a lot of people with like regular diseases and unlock like the next step to extending healthy lifespan.
Awesome.
Amazing. I agree on everything. No, just one comment. I think another issue is also the amount that you need
yes.
in itself. No? Because in the lab, we're pretty good in delivering factors. The problem is that stoichiometry in aging especially, the same factor expressed too much can cause cancer, but expressed in a small amount. So I think it's also a big challenge in the field that we are also stuck and it's difficult.
Yeah, definitely. Having precise control of dosage and duration is another key issue to making genetic medicines better. We'll know that different individuals will probably need different amounts, and then because we're going to get non-uniform delivery and the different cell types are different, like if you have like a reprogramming factor, how much reprogramming exposure you need to de-age like your hepatocytes is probably going to be pretty different than like your T cells and your hematopoietic stem cells. And being able to regulate that in a specific way, would also be super useful.